donor line in the background of IR24. Then in collaboration with CCMB, this particular program was coordinated by Dr. Ramesh Sonti, and from ICAR-IIRR, I led the team along with Dr. N.P. Sarma, Dr. APK Reddy, Dr. Gaurishankar Laha, and other readers like Dr. Shobha Rani, Dr. Viraktamat, and then other mentors were involved in this particular program. So when we started this particular breeding program, we were not sure. Usually backcross breeding program will continue up to BC6, BC7 or BC8 and will take at least 5 to 8 years for completion of the backcross breeding program. But the climate in Hyderabad was, the weather or climate in Hyderabad was very unique. We could take up at least about 3 seasons, or if not at least 2.5 seasons in a year. So what we decided is that, considering the premium grain and cooking qualities of Samba Masuri and also high yielding nature of Samba Masuri, we decided that we have to go for at least 4 back crosses. In the hindsight, I would say that this was not the proper decision. Probably we could have stopped at BC3 or BC2 generation itself. But at that point of time, there were very few papers which talked about mark-crested breeding or mark-crested backcross breeding. We decided that we have to get back the complete Samba Masuri plus the bacterial blight resistance. So we opted for 4 rounds of backcrossing. So in this process, our first set of crosses were made in way back in 1999. And by the year 2003-2004, we had BC3F4 lines ready for evaluation for the resistance and then yield and all. And by the year 2005 and 6, we had already BC4F6 lines ready for yield trials in All India Coordinated Rice Improvement Program. So in this program, as I informed you earlier, we used SS113, which is the NAL of PR106 as a donor for Exite 21, 13 and 5. And we used 2 recipients, Samba Masuri and Triguna. I'll be restricting my talk discussion only with respect to developments in Samba Masuri. We have also developed improved breeding lines in the background of Triguna. But let me restrict my talk only with respect to the developments in Samba Masuri. So we decided to use a set of markers specific for Exite 21. For example, we had a amplicon length polymorphism marker, which was very close to the gene or part of the gene. For Exite 13, we used a CAPS marker. CAPS marker are also called as cleaved amplified polymorphic sequence markers or PCR based RFLP markers. So for using a CAPS marker, what you have to do is, there will be 2 rounds. The first round will be amplification using that marker, followed by restriction digestion with a specific restriction enzyme. For Exite 13, it was HINF1. So after restriction digestion, the resistant and then susceptible donors will show clear-cut polymorphism, and then we can identify which particular backcross plant is homozygous for the donor resistant, for the recurrent parent allele, and which one is showing heterozygous for that allele. So for Exite 5 also, it was a CAPS marker. And to accelerate the backcross breeding program, we also used a set of 90 parental polymorphic SSR markers. So we had at the point of time in 1999, there were a total of 350 SSR markers or microsatellite markers available in public domain or public databases. So we ordered all those markers, checked for polymorphism between SS113 and then Samba Masuri, and then we identified 90 markers to be polymorphic. As informed earlier, just to ensure to be on the safer side, we did backcrossing till BC4F1 generation. So this particular chromosome map describes where those genes are located. For example, Exite 5 is located on chromosome 5, and we have got a very closely linked marker or marker which is even can consider as part of the gene called RG556. For Exite 13, the marker was about 1.3 centimorgan, and for Exite 21, the marker was part of the gene. It was called as PTA248. And the ones given in green color, like RM6340, these are all the markers which we use for background selection in order to accelerate the recovery of Samba Masuri genome. How these markers are helpful, I'll explain to you later. So this particular gel picture or electrophotogram describes how we did the selection at BC1F1 generation, wherein we had a set of around 145 backcross plants derived from the cross between SS113 and then Samba Masuri. The other name for Samba Masuri is BPT5204. If you can see here, this is a clear-cut polymorphism shown by the marker. After PCR, when you load it in a gel, all the plants which have the resistant allele, either in a homozygous or heterozygous condition, will show a 900 base pair fragment or almost 1 kb fragment. And all the plants which are susceptible will show a 400 base pair fragment. As you can see here in this particular gel picture, we have plants which are either heterozygous for the target gene or homozygous susceptible. So you'll not get a plant which is homozygous resistant because this is BC1F1 generation. So in this particular gel picture, as you can see here, plant number 276, 274, 273, 271, etc. These are the plants of interest to us because it had the resistant gene in a heterozygous condition. So this DNA isolation and then PCR was done with 15-day-old seedling. So on the same day, we identified that about 44 plants were positive for XA21. Then those 44 plants, the DNA was once again taken up, and then PCR was set up for XA30. After PCR, we did restriction digestion with the restriction enzyme HINF1. So with respect to XA13, as you can see here, the resistant parent shows a 400 base pair fragment, which is specific for resistance. Samba Masuri or BPT shows a 1 kb fragment. So we are interested in identifying plants which are heterozygous for the resistance specific fragment. So we got about 23 plants which were positive for XA30. So these 23 plants are not only positive for XA13 but also for XA21. Similarly, these 23 plants, we subjected them for marker state selection on the same day for XA5 gene. About 11 plants were positive for XA5 gene. This process of marker state selection was repeated in all the 4 generations, wherein in case of BC1 F1 generation, out of 145 plants, we got 11 plants which were triple heterozygotes or triple positives. In BC2, we got 9. In BC3, we got 12. And then BC4, we got 10 plants. But we were in a dilemma. We have about 11 plants, like say, for example, 359, 360, 361, 273, etc., which are all positive for all the 3 target genes. Which of these plants to use for backcrossing? Theoretically, we can use all these plants for backcrossing, or ideally, I'll look for some of the phenotypic attributes among these backcross plants, which are similar to Samba Masuri, and then I can select a particular plant. But at that point of time, it was through a review paper published by Hospital and Charcoset. They told that if we have a codominant markers like SSRs, we can use them for background selection or selection for Samba Masuri genome, so that we can accelerate the background genome recovery through with the skill sets of CCMB, wherein they had a sequencing or capillary electrophoresis system, wherein at least about 7 or 8 SSR markers differing in size ranges, we could resolve them. So in this particular gel picture, we have a set of 11 plants as listed here, 359, 360 and all. So they were identified, or they were subjected for background selection here. As you can see here, this is a selection with the help of 2 markers. A small section of gel picture is shown here. We did it with all the 90 markers, but if my selection among these 11 plants, if I had to make a selection for advancement to the next generation, which plants I'll select? Definitely I'll not select a plant number 1 and 2 because they are still heterozygous for Samba Masuri allele. I'll certainly select plant number 3 and 4 because they are already homozygous for Samba Masuri allele with respect to RM90 and also with respect to RM201. Similarly, plant number 7 and 8 are homozygous for both RM19 and then RM201. These are SSR markers located on different chromosomes. So like this, we did it for all the 90 SSR markers, then identified how much is the recovery with respect to background genome. So for example, in the best plant, that's plant number 273 and BC1F1, through background selection, we identified that in that particular plant, plant number 273, 65 markers out of 90 were homozygous for the recurrent parent allele. This was, like say, not very near to the theoretical value, but near. Ideally, we expect that 75% of all backcross plants will be similar to the recurrent parent. And by BC2F1, we had a plant which had 80% recovery. By BC3F1, we had a plant which had 89% recovery. By BC4F1, since we had added additional number of plants for background selection, we had a plant which had 96% background genome recovery, which means out of 90 polymorphic markers, only 3 markers were heterozygous. All the remaining markers were homozygous. So this particular process of marker-state backcross breeding was repeated till BC4F1 generation. And then when we sell the BC4F1 plants to BC4F2 generation, we had a set of 5 3-gene pyramid lines, which had all the 3 genes in a homozygous condition. and they were subjected for evaluation for the morphology, yield, grain quality, and also bacterial blight resistance. Fortunately, all the 5 homozygous lines were resistant to or highly resistant to bacterial blight. But we selected, since there was a limitation in the number of lines which you could nominate, we selected 4 best lines, which include IET 19026, 19045, 19046, and 19590. They were nominated for All India trials or All India Coordinated Rice Improvement Program trials in the year 2015 and 16. So they were tested very vigorously in bacterial blight hotspot locations, both for their yield and also for their grain quality and also for their bacterial blight resistance. Finally, one of the entries, IET 19046, was selected and then identified for release as a new variety. As you can see here, in all the hotspot locations, in both artificial and the natural inoculations, this entry, IET 19046, which is an improved version of Samba Masuri, having all the 3 bacterial blight resistant genes, it showed excellent immune level reaction to bacterial blight. And it also was very similar to Samba Masuri in all the agromorphological traits except for maturity, wherein IET 19046 matured about 7 to 10 days earlier. Based on all the superior characters of IET 19046, it was identified and then released as a new variety or a NAL of Samba Masuri called Improved Samba Masuri. And this was also registered with PP and FRA of Government of India. What is unique here is that 2 institutes, one institute ICAR-IIRR, which is a institute working primarily on rice breeding and then development of rice technology, and Center for Cell and Molecular Biology, wherein the group was headed by Dr. Ramesh Sodhi. We both partnered, and then we were able to develop and then release a product for public good. And as on today, Improved Samba Masuri is slowly but steadily replacing Samba Masuri out of the total area of almost 3 million hectares under Samba Masuri. So it has very modestly but steadily spread to all bacterial blight endemic areas. As on date, anywhere between 2.5 to 3 lakh hectares is under occupation by Improved Samba Masuri. So initially, our main problem was that we were not able to produce enough seeds of this variety and then distribute it to farmers. So we licensed as a production of seed to 2 private companies. One is Sweet Biotech, another is Metahelix, which is part of Tata Rallies group. And then we were able to increase the area. Right now, as on today, it's about 2.5 lakhs to 3 lakh hectares. And in between, when we subjected all our rice varieties developed at Indian Institute of Rice Research for the glycemic index. So one surprising discovery or serendipitous discovery was that Improved Samba Masuri had a very low glycemic index value of 50.99. In general, rice varieties like Swarna and other rice varieties have a glycemic index anywhere between 62, 65 or 70%, which means that if a person eats those varieties, at least 60 to 65%, it will be immediately released as glucose, and then it will result in shooting up of the blood glucose levels. But this particular one, I'll show you the values and other things later, had very low GA values. So because of this particular low GA value, as on date, at least 3 major companies like KRBL, then Adani Wilmar have shown interest to grow this variety and then market it on payment of royalty. And CSIR has also helped us in giving a small funding through a project called as Blightout, wherein we have managed to distribute seeds of this variety to more than 10,000 Samba Masuri farmers in several rice growing states in the country. Because of this particular development, our team also received various team and then individual awards. And this is the way the crop looks like. This is captured from a farmer's field, Improved Samba Masuri. And we also published a paper highlighting how the marker-stud selection or marker-stud breeding process was done. If anybody is interested, I'll be happy to share, wherein we have described our journey in terms of development of Improved Samba Masuri. And what is important to note here is, it just like Samba Masuri, Improved Samba Masuri also commands the same premium market price, which means if Swarna gets about 1,800 or 2,000 rupees per quintal, Samba Masuri or Improved Samba Masuri will get around 2,300 or 2,400 rupees per quintal. And in severe disease conditions, for example, after the cyclone Hudhud, which happened in coastal Andhra Pradesh or near Visakhapatnam and then Kakinada, Samba Masuri was totally gone. As you can see here, because of bacterial blight, this is totally gone. And Improved Samba Masuri, as you can see here, it has already started to mature and then it's showing full grains, not even a single speck of disease. This is where resistant breeding has really come into picture. An independent survey carried out by National Academy of Agricultural Research Management they have published a paper independently, and they have shown that because of its bacterial blight resistance, it has certainly added value to many bacterial blight prone areas, wherein farmers are able to get 25 to 40% more yield than Samba Masuri. And this translates in just a 5-year period a benefit of more than 240 crores. Remember, for this particular project, the funding was only around 50 lakhs, of which 22 lakhs came for ICAR-IIRR and then 30 lakhs came for CCMB, with just funding of around 55-60 lakhs. And then with the help of our acreage system, we evaluated this particular variety. Now see the benefits which biotechnology has helped in accruing. This is for a small period of 2011 to 2016. If we do a fresh survey, probably this value will be around 500 crores. So I'm showing this slide just to show that biotechnology is indeed rewarding, and then working in biotechnology will be contributing a little bit towards improving the livelihood of farmers. And as I told you earlier, the grain and cooking quality of Samba Masuri and improved Samba Masuri are almost equivalent, or if not better. And this is a recent development wherein NAS, a National Institute of Nutrition, which is the only testing agency to certify for glycemic index through clinical trials, they have estimated that improved Samba Masuri has glycemic index of about 51%. And as I told you, consumption of low glycemic index rice varieties like improved Samba Masuri and other varieties will be very good for patients suffering from type 2 diabetes. And we have also worked on characterization, because we were not happy with just deploying Exate 13, 5 and then 21. We also wanted to know how much time it may take for defeating this particular resistant gene combination. To our surprise, this was a study done in 2015 to 2017. One of my PhD students, who is working with Dr. Gauri-Shankar Laha, he has done this study wherein more than 800 isolates were characterized. They belong to all the rice growing regions in the country. As you can see here, pathotype 1 to 15, 16 are not the ones which are dangerous. Pathotype 19, 20, 21, 22, particularly 21 and 22 are appearing to be very dangerous. As of now, they are limited to small areas in Assam and also in Faizabad. But probably once this particular variety will be deployed in the next 5 or 10 years, probably it may result in breakdown. And as you can see here, 5, 13 and 21, it's showing severe resistant or moderate resistance only. Even improved Samba Masuri, one of these isolates, is showing only moderate resistance. Still, you cannot say it as compatible. It's moderately compatible only, we can say. So there is certainly a need to identify novel sources of resistance. My research group has also worked along with other research groups in CCMD, Indian Institute of Institute for Microbial Technology, and then characterized bacterial blight isolates from different parts of the country. And we have also worked on identifying novel source of resistance. And I'm proud to say that this is one of the first resistant genes identified and then cloned in the country. This gene, called as Exate33, has been identified from Moraiza nivara, and we have already transferred this into the background of several varieties like Akshay Dan, Samba Masuri, and then improved Samba Masuri. And we have also identified a major recessive resistant gene called as XAAJ2, which can also be called as a recessive QTL, which can contribute to about 30% of phenotypic variance. And this is one of the most recent developments. Parallelly, the research group led by Professor Kuldeep Singh, who is presently director of NBPGR, they had identified another major resistant gene called as XA38 from Moraiza nivara, a wild rice. And then this gene was transferred to the background of Samba Masuri and improved Samba Masuri. This year, an entry of Samba Masuri possessing, in addition to XA21, 13 and 5, XA38 has been found to be promising, and then it will be shortly released for 7 rice growing areas in the country which are endemic to bacterial blight. And then important thing is that it gives at least 12% more yield than improved Samba Masuri, and also gives better level of resistance, which means when the pathogen is actually slowly developing compatibility against Samba Masuri or improved Samba Masuri, we are also working towards developing more durable resistance. But one important point what we need to understand is we have certainly taken care of bacterial blight, and we are developing or adding additional resistant genes so that we can make resistance in Samba Masuri or other varieties more durable. But what we need to understand is most of the popular varieties, be it Swarna, Samba Masuri, MTU-1010, Pankaj, all these varieties are susceptible to other major diseases and also pests and also abiotic stresses, particularly drought, submergence, and then salinity. So we decided way back in 2012-13 itself, we were not happy with just developing improved Samba Masuri and then basking over the laurels. We wanted to add or fortify or add value to Samba Masuri by adding additional resistant genes, 1 or 2 genes for other stresses. So where we are with respect to this particular effort, I'll be in the next part, a few slides, I'll be talking about development and evaluation of breeding lines of improved Samba Masuri and other varieties possessing resistance against multiple biotic stresses and also abiotic stresses, and also how we have also managed to add an important trait, that is yield improvement, in the background of Samba Masuri. So this is presently what we are working on. As I told you, for bacterial leaf blight, we have already added XA38, XA33, and then XA-AJ. And XA38 product is already ready for commercial release, while products in the background of improved Samba Masuri with XA33 and XA-AJ are in various stages of development. Similar is the case with blast resistance also. We already have products with PE2 and PE54, which are there in the final year of testing. We have also added BPH resistance into the background of Samba Masuri, wherein a new gene, a very broad spectrum resistant gene called BPH33 has been added. We also added another pest resistance trait called as Galmage resistance, wherein we have added genes like GM4 and GM8. And we have also added low soil phosphorus, salinity tolerance, and then drought and submergence. Low soil phosphorus and then salinity tolerance are already ready for commercial release. So, for example, if we can, earlier I showed that improved Samba Masuri is highly susceptible to blast, which is very much evident from here. But by addition of 2 genes, PA2 and PA54, the lines are showing very durable resistance. And one of the lines is in the final year of testing. Probably next year it will be released as a new variety. And this effort, I'm working along with my colleagues in pathology, Dr. Srinivas Prasad, and also my colleague, Dr. Seshu Madhav. And I also told you that BPH is becoming a major problem in many rice growing areas in the country, particularly north, east, and then southern part of the country. So now we have a version of Samba Masuri fortified with a new BPH resistant gene. This is a gene which we have identified, cloned, and then characterized. This particular gene, the candidate gene, is a gene which encodes chaperonin protein or a heat shock protein, and it works very efficiently. So when we tested improved Samba Masuri line possessing this particular gene at different locations, as you can see here, in all the BPH prone areas, it's showing very high level to moderate level of resistance, not only to BPH but also to another pest called as WBPH, which is also called as white bank plant hopper. And same is the case with the Galmage resistance. Here, what we did is that we didn't go for 4 or 3 generations of back crossing, just 2 back crossing and then further generations of selfie. We were able to identify lines of improved Samba Masuri with more number of grains or bunchy panicles also. So these lines have entered all India trials, and we have also tried to add an additional trait, tolerance to a major recalcitrant disease called as sheet blight, through a population by crossing an important donor called Vazopec and then improved Samba Masuri. We have developed a set of RILs with same grain type like improved Samba Masuri, possessing a major QTL called as, we have not yet named this particular QTL. A major QTL we have identified from Vazopec, which can confer very good tolerance to sheet blight. So we are trying to incorporate it through backcross breeding and then selfing into the background of improved Samba Masuri. And another effort, as I told you, is low soil P tolerance. You may be knowing that majority of rice growing soils, particularly the upland rice and also a lot of problem soils like acid sulfate soils, hilly regions, they are all having a very low to low and then moderate soil phosphorus content. Even if soil phosphorus is there, it will be fixed in from available form to unavailable form. So way back in 2002 to 2011, International Rice Research Institute, working with Indian scientists and also working with Japanese Agency for International Development, that is JIRCAS, they had identified a major QTL called as POP1. Later, with collaboration between International Rice Research Institute and in University of California, Davis, and then also scientists from Japan. They identified that particular candidate gene. It was a serine threonine kinase called as PS-tol kinase, and the gene was identified and then cloned. What is unique to know is that PS-tol kinase was cloned from an Indian landrace called as Kasalat, which is still being grown in several parts of eastern India. And what does this QTL do? Under normal soil, this QTL will not produce any effect. But under soils with low soil phosphorus or in upland soil, this QTL detects lack of phosphorus and then it will produce more roots, as you can see here. The variety which doesn't have popcorn, like Samba Masuri, it will not produce sufficient roots. While with popcorn under low soil P condition or low water conditions, it will produce more roots and then trying to tackle or survive under low soil phosphorus conditions. So IRRI had already transferred it in the background of IR72, IR64, and then varieties like Siharan. At ICAR-IIRR, we have transferred it to the background of improved Samba Masuri, MTU 1010, IR64, and other rice varieties. And already products have been tested.